Good morning, everyone. It's Ed here. Welcome to BIYSC. Today is the end of the program. First and foremost, thank you, all the participants, researchers, and friends for joining us today. Your dedication and enthusiasm have shaped this unique journey, and it's truly an honor to celebrate your achievements together. Over these days, you have immersed yourselves in the spirit of scientific exploration, challenging assumptions, collaborating across cultures, and pushing the boundaries of knowledge. Your commitment to rigorous inquiry and creative thinking has not only produced remarkable projects, but also has forged lasting friendships and vibrant community of young scientists. Today, as the program draws to a close, we turn our attention to you, the real protagonists. We are eager to discover what you've learned, to listen to your presentations, and to be inspired by your vision of the future. The fruits of your curiosity and perseverance are not just a milestone for yourselves, but signals of hope and progress for us all. You stand at the forefront of a generation ready to tackle the challenges of our world, be it climate change, new frontiers in medicine, or the mysteries of the universe. I'll keep my remarks really brief today, so we may fully appreciate your accomplishments. The stage belongs to you, and we are all here to celebrate your passion and talent. In the spirit of scientific perseverance, let me leave you with the words of Marie Curie. Nothing in life is to be feared. It's only to be understood. Now it's a time to understand more so that we may fear less. May this wisdom accompany you as you continue to explore, discover, and make an impact through your curiosity and dedication. Thank you for your energy, your brilliance, and the promise you bring to the future of science. Congratulations to you all.
Thank you very much, Luis, and hello everyone. Today is the day. It doesn't look real, but it's already the closing ceremony. Over the past days, you guys have poured all your energy, good vibes, and hard work in these inspiring projects, demonstrating this hard work, creativity, and curiosity. And now is the moment together to celebrate this achievement, but also to listen to the results and the insights that now you're ready to share with us. We are truly excited to listen to your presentations. So once again, thank you for these amazing shared days. Congratulations for all the hard work you've poured into this. And now it's the moment. The stage is yours.
Hi everyone, I'm Caral Nomen, the staff member of the Quantum Cryptography group from ICFO. I want to thank everyone for coming, especially to my 8 students that are the best. They are brilliant, original, and so funny. I had so much fun with you. Thank you so much. And please give a big applause to them. You have to. And please come here, Grace, Xavier, Pablo, Gleb, Cindy, Victoria, Emma, and Xavier.
Good morning, we're the Quantum Cryptography group at ICFO, and these weeks we've been looking at the mathematical and physics foundations of this technology and applying it in experiments and programming. Moreover, we had the opportunity to visit the Cybersecurity Agency of Catalonia and to spin-off companies at ICFO. We've really enjoyed this experience, so we've made a video capturing the main ideas. We hope you like it.
up, bro. We're late.
Maybe they could help. Guys, I really like Alice, but I don't want Eve to know. Any ideas?
You could use the RSA encryption.
I mean, she could figure out the message, and I would have no way to know.
Use quantum cryptography instead. The BIYSC port is perfect for sharing a key secretly.
What's that?
Basically, Alice sends photons, which are particles of light that have quantum properties.
Then she polarizes them in a random basis, a rule book for encoding zeros and ones. This means she's changing the direction the light is vibrating in.
Like horizontal and diagonal?
Yes, and you measure them by using your own random basis and get zeros or ones.
Yeah, and as you can see here, here are the most popular bases, the computational basis and diagonal basis. The computational has vertical, horizontal, diagonal has diagonal and anti-diagonal.
Yeah, and then the only thing left to do is share the bases and keep the bits where these bases match.
Oh, and that becomes the secret key.
Yes, and if Eve eavesdrops, you'll know.
Because when you measure light, you might get a different result since the stage collapses. And this result may differ even though you have the same basis. The same happens when you try to copy photons. So basically, she'll never catch you without you noticing.
Genius, let's try it out.
Let's meet me tonight.
Yes! This quantum cryptography stuff actually works.
Hey girls, I need to talk to you about something ASAP.
OMG, what?
Bob's been recently acting very weird. He's always on that device, grinning and giggling.
Oh my god, girl, he's probably cheating. Break up with him right now.
Yeah, Heike, I saw him building like a weird machine.
OMG, what?
Yeah, look.
Babes, I think that could be a quantum cryptography machine.
Queen, what the heck is a quantum cryptography machine?
It uses photons of energy to transfer messages using bits. In order to eavesdrop, you need to be able to guess the bases that Bob is using, but be careful, because they can tell if someone's interfering with their message.
Oh my god, slay, period. I'm so hyped to try it out. I'll let you ladies know how it goes.
Sounds good.
Bit 1, I used horizontal. Same, keep it. And for bit 2, diagonal. I used horizontal, discard. Wait, bit 5 wrong. Someone's listening.
It was me. I spied and got you, Bob.
Is she your girlfriend?
Why are you texting her?
He said he was single.
He's literally my husband.
BB-84 show an eavesdropper, because you are a liar.
The mismatched bits gave you away, Bob.
Wait, I can explain. No chance.
Come on, he's such a loser.
Hello, everyone. So I'm sorry, guys, but Amanda is not here today. So I'll present the group of Josep Carrera's Leukemia Research Institute. So please come to the stage.
No te preocupes, no te preocupes.
Or you can speak there.
Good morning, everyone. We are the multi-omics in the fight against leukemia team. So, what is leukemia? Leukemia is a cancer of the blood and bone marrow. And one of its most aggressive forms is acute lymphoblastic leukemia, or ALL. ALL is a type of leukemia that affects lymphocytes, a type of white blood cells. Our team focuses on using multi-omics tools to help us better understand and fight this. Oh my God, help, is there a doctor in the room?
We have a 305. What happened, sir?
She has leukemia, but she's been taking her pills.
Oh, these pills are an analog for the gene P10.
We have our two suspects here, genetics and epigenetics.
Genetics, you are responsible for the genes which code the information in the cells.
Epigenetics, you play a role in the gene expression.
Let's take a blood sample and sequence the P10 gene. Oh, the P10 gene isn't working right.
Is it mutated?
It is, but it's just a polymorphism.
Next, we should check the expression of the P10 gene.
Is it expressed?
No, it's not expressed.
So if I'm not expressed, then my mutation, my polymorphism doesn't matter.
I mean, I silenced the gene because I said there was a mutation in it.
Didn't you think that silencing the gene would make the cells divide uncontrollably?
Oh, my bad. I didn't think about that.
What about methylation? Was she methylated?
Yeah, of course I did.
She was 77% methylated.
This means she's hypermethylated.
That's why the gene wasn't expressed. You killed her.
No, I did not, because then the drug should have worked.
So strange.
Precisely when the P10 gene isn't working right, the drug should take its function and save her life.
This is when medicine stops and then.
Each year, over 25,000 Spaniards hear the word leukemia for the first time. And
while 60% survive, the other 40% doesn't make it past 5 years.
Behind each statistic, there's a face, a family, and a future. With the lab coats. You were good, don't worry. That was a good transition.
Perfecto. Tengo el punto aquí, ¿vale?
Very good.
Thank you. ¿Necesitan?
Sí. ¿Em deixes els dos? Ara els hi dono. Sí.
Okay, so hi, good morning. I am Sarah, the staff member of the Drosophila and tumorigenesis group from IRB, and this has been my first year as a staff member, and I wanted to congratulate them on their project. So please come here and explain it to everyone. To all of you, Binming, Matilde, Joseph, Mayal, Mia, Olivia, and Sofia. Thank you.
Okay, hello everyone. Our project is unraveling tumorigenesis and its pathway Drosophila melanogaster as a model organism. It's a lot. Over this past week, we've used flies to model and create and study tumor growth.
Why do we use the fruit fly? We use this fly because they have a short life cycle, reproduce quickly, and have a simple diet based on cornmeal and yeast. Also, they share about 70% of their genes with humans, and their genomes are really easy to manipulate, making them an ideal model for genetical research.
So what do we mean by easy to manipulate? The UAS GAL4 system allows us to control gene expression within Drosophila. By crossing GAL4 driver flies with the UAS target gene flies, we generate offspring where GAL4 proteins activate the gene of interest.
In our case, we use this system to interfere with the spindle assembly checkpoint, which leads to an abnormal number of chromosomes in the cell, so cell death would occur. However, our additional introduction of the P gene 35 blocks this mechanism, leading to uncontrolled cell proliferation and tumorigenesis.
We began our experimental process with the extraction of the control 7-day and 10-day larvae. After dissecting the chosen larvae, we performed repeated processing of fixing, blocking, and washing to ensure tissue integrity throughout our procedure. Next, we applied the primary and secondary antibodies, which included the fluorescent dyes needed to distinguish our tumor cells from the surrounding tissues. After antibody staining, we proceeded with mounting wing discs and fat bodies. Then, these samples were later screened through a confocal microscope, where we processed and analyzed the resulting images.
In this experiment, we're inducing the tumors in the red area. When we look at tumor progression, we look at a 10-day period. We observe at the first control, we see that the tissue is normal and the cells are organized. But by 7 days, we see an increase in the red, which shows the evolving tumor. But by 10 days, we see a massive increase in the tissue overgrowth with a final occupancy of over 90%.
Additionally, tumor cells gain high mobility and they invade nearby tissue. In the control image, we can see a very well-defined border and all the red cells in their corresponding region. Whereas in the other image, we can see that the tumor cells have already invaded nearby regions and has caused a very disorganized border. In the zoomed image, we can also see how two tumor cells are in the process of migration with their membrane still attached to their original tissue.
However, tumorigenesis can be reduced with the use of puckered. Puckered is a protein that inhibits the oncogenic pathway, J and K, by preventing excessive signaling of this cascade. In the tumor condition at 10 days, the growth of the malignancy is evident, with increased MMP1 shown by the green region, suggesting tumor invasion and progression in the region. On the other hand, the rescue condition shows significant tumor suppression, returning the tissue to a near normal state and drastically decreasing tumor occupancy percentage and size.
But does the tumor affect other parts of the body? In these pictures, we can see the difference between the lipid dispersion of the fat body tissue in a control animal versus an animal harboring a tumor. Because tumor cells proliferate uncontrollably, they require larger amounts of energy. This is why the control animal has a normal amount of lipids present among the cells, while on the right we can see that most of the lipids have been mobilized and used.
Finally, we use CRISPR-Cas9, a new genetic tool that allows us to directly edit and cut DNA. CRISPR consists of two parts, the Cas9 that cuts DNA and the gRNA that recognizes the target. We use CRISPR to change the fly's natural red eye color from the wild type into white eye color. The whiter the eye, the more effective the CRISPR. 99% of our flies had a partially white eye, proving the value of CRISPR.
Thank you.
Thank you.
Hello, I'm Laia Shaw Planas, and I'm going to introduce you the group from ICIQ. It's been a pleasure working with you guys. You're all very passionate. So let me introduce you this group. Nicholas, Angela, Ella, Arthur, Arina, Maria Paz, Martina, Leo, Kavi, Natalia, and Mateo. The floor is yours.
We can all agree that the world is currently facing many problems.
Such as climate change.
And the solution to these problems is
chemistry.
We've been working on dye-synthesized solar cells, DSSC, which is a type of cell that uses dye to absorb light. This concept is inspired by how chlorophyll works in plants. In these cells, light excites electrons in the dye, producing a current. A copper electrolyte returns the lost electrons, thus completing the circuit.
Here's how we made our cells. We started off by cleaning the electrodes. We then applied titanium dioxide to one electrode. This absorbed the dye, which we extracted from curry and beetroots. Next, we coated the other electrode with a layer of graphite to increase the conductivity. And the final step involved assembling the cell and inserting the electrolyte between the electrodes. Upon testing, our cells proved successful.
Solar cells are an ideal source of renewable energy, as by harvesting just a small amount of incident light, they can provide significant power. In addition, DSSCs are able to absorb a larger range of wavelengths of light compared to other solar cells.
Moreover, DSSCs are especially promising, eco-friendly alternatives to the current silicon models. DSSCs can play a crucial role in the production of fully green hydrogen by using electricity generated to power clean hydrogen at water-splitting reactions.
Making hydrogen production sustainable should be greatly prioritized as it is an energy carrier with the potential to be used in transportation, heating, and power generation. This would greatly reduce greenhouse gas emissions. Therefore, green hydrogen can revolutionize the renewable energy market.
Green hydrogen is produced by water splitting. This process uses electrolysis through tap water, which is an electrolyte solution, to produce the endothermic reaction. Through this, we split the water and we obtain oxygen and high levels of hydrogen. So hydrogen has also some limitations. So research in a performing catalyst is very crucial to accelerate the rate of the reaction and therefore lowering the supply of energy required for splitting the hydrogen, like the water. There's also another limitation, which is the most important one, is that hydrogen is a very explosive element. So research to reduce the pressure exerted when conserving this gas will deal with the frightening myth of explosion. So like BIYSC shows us, we need to invest in innovation for a better tomorrow.
And finally, drugs. You love them, we made them. But how do we make them? One technique we used is called retrosynthesis, which is where we start with our target molecule and then we work backwards by tactically splitting it up into what we call synthons.
Now, synthons are like the puzzle pieces of our molecules. They don't actually exist, but we can use them as foundations to find real molecules that will react to get to our target. Now, we actually use this ourselves to make sodium acetate, as you can see on the right-hand side, and sodium acetate is a common shelf life extender used in foods.
Medicinal chemistry is all about designing and developing new drugs. Having progressed from herbs and alchemy, the industry is now worth over $1.8 trillion. The desired hit substance has sufficient activity against disease is found, and then it's adapted to optimize its structure. Finally, it undergoes a long cycle of clinical trials before becoming commercially available.
This is a paracetamol molecule. It is found in painkillers such as Tylenol or Dafalgan, and we synthesized it. Through reverse synthesis, we got those two synthons, which can be obtained from acetic anhydride and p-aminophenol. Using these, we synthesized some crude paracetamol, which we then purified and tested. And although it isn't quite Walter White level, it's still pretty good. Thank you for listening.
Hi, I'm Bru. I'm the staff member for the infectious disease group, and I truly have no words to describe them, but just please give them a big applause because they are such a group with brilliant minds and hard, kind, and empathetic that are really changing how we see medicine and HIV care. So please welcome to the stage Faisal, Felipe, Valeria, Alina, Elena, Laia, Renata, Trinidad, and Cristina.
Hello everyone, I'm Faisal.
And I'm Christina.
Together, we're from the infectious disease group. Over the weeks, we've been working on HIV and STI at Hospital Del Mar.
These are our 9 most important learning outcomes that we have reflected on during these past 2 weeks. We also focus on what it's prevention, treatment, patient care, research, and diagnosis. As a result, we made a little video to summarize what we did, and hope everyone enjoys it. Thank you.
Hello Valeria, how are you?
I'm okay, I'm kind of nervous, but okay.
Could you remind us why you came to see us last time?
I had a fever and my body was aching, but the fever has gone down recently.
That's good.
So we have the results back and you've tested positive for HIV.
What does that mean?
HIV is a virus that affects your immune system. Specifically, it affects some cells called CD4 cells, but it's not really important. What is important is that there's very good treatment for it, so there's no need to worry. Felipe will tell you about the treatment plan in a second, but what is important is that once you become undetectable through the treatment, you won't be able to transmit the disease to anyone.
You shouldn't be nervous. The antiretroviral treatment is very effective, and there's many ways you can take. You can take it as a pill a day or as a shot every 6 months. You should tell us what you prefer.
I think definitely a pill a day. I'm kind of scared of shots.
Okay, that's fine. Do you have any other questions for us before we start in the treatment?
Will I be able to have children?
Yeah, you can have children. As I said, once you become undetectable, you're untransmissible. That also counts for if you have a child. The only thing to take into consideration is that if you do want to have a child, maybe consult with a doctor because there's some risk with breastfeeding because it might be possible to transfer the disease through breast milk.
And can I still have sex, or?
Yeah, obviously. The most important thing is like you still need to be protected every time you have an experience with another person, so you don't transmit another STI.
I'll give you this information pamphlet. There's some more information about the disease, and a nurse will call you to tell you how to maybe change your habits so that you feel better.
Okay, thank you guys. Hey guys, I have some news to tell you guys. I went to the doctor not long ago, and I tested positive for HIV. I started treatment not long ago, and I just thought, like, as my friends, you guys should know, because I've been having like a rough time like dealing with it. So yeah.
That's disgusting. Yeah, that's like really gross.
Don't say that, like, don't blame her. Because HIV is just like another infection, and you're not gonna be sick just being here.
But I don't want to be around with like someone like that.
Yeah, but that's not how HIV is transmitted. It's not transmitted through touch or like on the air. No, it's through bodily fluids. And plus, she's on treatment, and the treatment makes you undetectable. And if you're undetectable, you can't transmit the infection.
And like any other disease, it could be like stopped. There are 4 simple ways that you could prevent getting HIV, such as just using protection, or if not, just go on PrEP. And just getting tested, it's cheap and it's for your own safety and the safety around you. And most importantly, if you do have HIV, just get the treatment so then HIV doesn't spread.
I guess that makes more sense. I feel more comfortable now.
Yeah, me too. I didn't know that.
Yeah, I'm sorry for judging you. I think I get it.
Yes.
I get it now.
Yeah, we love you, girl. We're here for you.
We're here for you.
Take the picture now.
Whatever. Hello, everyone. I'm Carla. I'm the staff member of the Cosmic Rays group at IFAE. And it's been really great to have you guys throughout these 2 weeks. I think the group dynamics was really cool within you, with me, and also with the researchers. So please welcome to the stage Pau, Aitor, Theo, Lera, Vika, Sanki, Ryan, Ignacy, Max, and Camila. And Timo.
Hi, we are the Cosmic Rays group, and we will present our project. First, cosmic rays are particles that travel across space at nearly the speed of light, and they hit Earth every day. Most of them don't go through the atmosphere and the magnetic field, but the ones they do, they collide with other particles and atoms, creating particle showers. That means that from the interaction of, for example, 2 protons, lots of different particles are created. Some of them are the muons, which we can detect with our devices.
As Nat said, muons are created by collision in cosmic rays. That's because muons are fundamental particles, which has some different properties that when there's collision and high energy ones, they can be rearranged and they can create different other fundamental particles. Muons are just heavier electrons, which means that their half-life is very short, it's 2.2 microseconds, but due to the integration and creation of other particles and other reasons, they end up arriving at the ground, and that's when we detect them.
As we all know, electrons have negative charge. As we said before, muons are just heavier electrons. Therefore, when muon goes through an atom, they repel each other because negative charge repel. And because electron has a much less mass, it becomes excited and moves to high energy state. But it's very hard for electron to maintain its position there because it requires a lot of energy. Therefore, it's going to move down to its original state. And during this process, the photon is released. And our detectors senses this photon.
This emission of light allows us to detect the muons. For the light emission, we've used this material, which basically is a material that emits more light than others, which is called a plastic scintillator. The plastic scintillator, we've placed it on top of a silicon photomultiplier, which is basically a light detector. So this is our muon detector. And what we've done is that we've placed two detectors, one on top of each other, because muons don't interact a lot with matter. So they pass through like with no problem. So when a muon passes through our detector, we detect two spikes of energy. Whereas if there's a random error or another particle passes through, we will only detect one spike.
So we can now detect some muons and actually do something about it. So first we propose that there might be a specific region in space that might have a higher amount of cosmic rays. So which means that the time of the day can actually affect the muon flux that we detected. However, we can't see any correlations, which means that the cosmic rays are actually evenly distributed across the universe. And the next thing we propose is that the location or the environment of the detector can actually affect the rate of muons being detected. So what we did is that we test the amount of muons detected outside the building, and we can see that outside the building there's actually more muons being detected. And thirdly, we also changed the angle of the detector and see how does it affect the muon flux being detected. So when we face it upwards, we can get the most muons, but however, when we place it sideways, we actually get the least number of muons, which can prove that cosmic rays or muons are actually come from the sky instead of other places. So in conclusion, throughout the whole program or the project, we learned some theories, we made the detector, we detected some muons, and we played some ninja as well. That's all. Hope you guys enjoyed the presentation, and hope you guys can find the penguins being hidden in the PowerPoint. And thanks for listening.
Good morning, everybody. I'm Nuria. I'm the staff member of Engineering Gaudí, Inhabiting Sound, Light, and Space. And it's been from Lab to La Pedrera. And it's been a pleasure staying with you. So welcome to the stage, Zihan, Yichen, Omar, Neus, Eric, Jamal, Stephen, Alexander, and did I miss somebody? No, I think everybody's in. Welcome to the stage.
Només en parlàvem.
Okay, good morning everyone. This is the group. Everyone's worked in this presentation. I'm just going to present it and let's begin. So, the Gaudí project, we're going to start with the big question which we established at the start of the project. The big question is, how can we apply the architectural and acoustic comfort of La Pradera to the future residential buildings? We began with the methodology by observing Gaudí's manipulation of sound, light and space. These are the three key aspects we examined. It was possible to conduct a research about the buildings and its perks. With the ISA lab and the help of both architects and engineers, we were able to explore the Pradera model for future residential buildings. Okay, so we split our time into the engineering side and the architectural side. With the architects, we spent Friday in situ in La Pradera evaluating the different soundscapes and areas from an architectural standpoint. This is evaluating the form, the flow, and how things are not only ornamental but have a function. And then this is just quickly showing where we visited around one of the plans. You also notice the plan is extremely irregular in shape. It's not quite uniform as we're used to. And then across our journey, we started off with 2D measurements, progressing to 3D, and 4D and 5D, which are a bit less well known. But 4D is essentially looking at the temporal, the people's feelings in the different spaces, and 5D is more of a examining the whole motif and contextual aspects of the areas. And then this is the engineering part where we look at the acoustics of each area. We essentially went around lots of different sites in La Pradera with a head dual mounted microphone, ambient link microphone, 360 video recording equipment, and we used the SPL sound meter to get the decibels, and we used the reverberation time to basically indoor to analyze how long sound is continued after speaking. We also conducted a lot of surveys in each situation, and then this is going to link into our lab measurements. In the lab, before we went to La Pradera, we spent a lot of time in the anechoic chamber, which is very, very quiet. It's crazy I wish you could have all been, but yeah. The reverberation room, where there's a lot of reverberation, up to 6 seconds. It's like the stairwell in the campus. And then we did a bit of work on the first day establishing the foundations. But the most important room is on the bottom right, the immersive room, which we used with the 360 video. We put it in the lab so we could assess in a different way how we perceive the sounds. And then here are some of the results I'm going to quickly go through. Analyzing the acoustic performance to comfort and assessment using technical and perceptual methods. Here's just a quick video, quick image to show us in different areas how it would look if you were there. And then we look at the roof. Here are the decibels of the roof. The numbers correlate to where we are in the area. And you notice how in the exterior there's a significantly higher amount of decibels, and as soon as you enter inside, even on the roof, there's a significant decrease. And then here is the inside rooms. We did some evaluations in there. Looking at the reverberation time, however, in the immersive room, it's always significantly higher because the immersive room naturally has a 0.4 second increase because of its configuration. Essentially, there's a lot of stuff from here, a lot of data, but the important thing is the immersive room works as a concept of listening to and simulating areas in real life and then in the immersive room. And then here is a little interesting case study of the bathroom compared to the courtyard. The bathroom is significantly smaller. Hopefully you can see the letter B on the image compared to the courtyard, which is massive, but there is a very similar reverberation time. And then if we look at the bottom right, this is a little graphic showing ideal reverberation times for different performances. For example, a speech needs a low reverberation time, whereas musical performance needs a high reverberation time to provide a fuller sound. In conclusion, the shapes of Lapidera improve the architectural and acoustic comfort. Ornaments help to diffuse the sound and achieve a better acoustic experience. An immersive room can be used with confidence to perform this kind of research. And thank you for listening. We appreciate everyone who's helped us with our goals, and everyone here has enjoyed it. And yeah, thank you.
Hi, now it's the turn of the Planarian group. Thank you for your work and your confidence during these weeks. Please give a huge applause to Camila, Elsa, Tina, Carlos, Ronin, Cesc, Aidan, Lena, Kitty, Jan, Andrea, Aina, and Yago.
So we've worked with Planarians, and we've worked with Planarians. And to begin, I'll explain what Planarians are. The idea of the flatworm species, and what's impressive about them is their regeneration. You can cut one into one or more pieces, and each individual piece will regenerate a whole Planarian. They do this because they have stem cells all across their body. And now my partner Jan will explain more concretely how they do it.
So it is thanks to neoplasts, these stem cells over here, which are pluripotent, meaning that they can regenerate into any type of cell. So this occupy like around 25 or 30% of the total amount of cells in Planarians. And this grants them this great ability. Humans, in the contrary, don't have this amazing capability due to the complex body form they have and their limited stem cell reserves, meaning that they can only regenerate the epidermis and some organs such as the liver.
Our goal is to explore Planarian regeneration limits, exposing them to irradiation or gene knockouts. The global aim of this is applying it to medicine. A good example would be paraplegics' spine or neural diseases recovery.
For our first experiment, we experimented with cutting the Planarians and observing how they regenerated over 7 days. In this one, you can see that the cut was quite deep, but we hit the wrong tissue, so it regenerated just in a single head. In the next slide, we can see that the cut was deep, wasn't as deep, but we hit the right tissue, so it regenerated 2 tails.
Okay, in these pictures taken on the 5th day of regeneration, we can see how the eyes and the head had begun to form in the control picture, but not in the gene X. And a week later, we can see that the eyes have all formed in the control one and not in the gene X, and that is due to the silenciation of a gene.
In that capture, we see in green the nervous system and in blue the cells, while when the gene CB-Pitch3 is inhibited, we don't see eyes nor brain. So we concluded that the gene's function was to differentiate stem cells.
Beta-catenin is basically a protein that activates a gene which makes the Planarian grow tails.
So what we've done is that using a gene editing technique known as the RNA interference, we are able to cut out the beta-catenin or inhibit beta-catenin from the Planarian. And we can observe that without the beta-catenin, the sample grow 2 heads after regeneration instead of how they're supposed to grow, which is 1 head and a tail.
So as you can see, these pictures we took from the microscopes, and these three are from the confocal microscope. And for these three, there's a normal one, so they have both the head and the tail. But for the inhibited beta-catenin ones, they have two heads instead.
Then we also made an experiment with irradiation. Irradiation is used in planarians to kill some of the stem cells so that then scientists can follow them more easily. So we had 3 different gradients of irradiation. We had a control group, the sublethal irradiation, and then a group with lethal irradiation.
Also, we used 2 different types of species, the Smithia mediterranea and the Drosophila tugrina. The lethal group claims to kill and exterminate all the stem cells in the organism and therefore kill the Planarian cells. But after our analysis, we concluded that Drosophila tugrina has a higher tolerance to that radiation because it had a lower tax of death.
All right, one way to study planarians is by injecting double-stranded RNA, which is used to silence specific genes. This is usually done by a microinjector. To practice this, we used a blue colorant instead of double-stranded RNA to analyze the digestive system of the planarian.
And here you can see 2 bits of machinery that we used in these 2 photos. This first one is, in fact, the microinjector, as she mentioned before, and this one is a micropipette puller, which essentially takes a filament and thermally separates it into 2 smaller needles, which we can use for the process, because you need really, really tiny needles to do this whole process.
As a conclusion, after a week of working with planarians, this experience has led us into the world of scientific research, particularly in regeneration. Additionally, this program helped us decide what we would like to do in the future, as well as introducing new concepts which broadened our scientific knowledge. Thank you.
Good morning. We are Julia and Aino, the staff members of the Hospital Clinics project.
We are immensely proud to present you our future doctors, Carolina, Madalena, Paula, Ona, Daniela, Anastasia, Akida, Ethan, Mariana, Matias, Yaretsi, Maria, and Jaura. The floor is yours.
Welcome all. This is the Browning Medical Expertise Group, and over the last couple of days, we have been rotating around Hospital Clinic and seeing all the different departments. On the first day, we visited the Imaging Diagnostic Center and the Biomedical Diagnostic Center. In the morning, we got to walk around with the residents, radiology and nuclear medicine residents, and they taught us how different techniques work, such as MRI, head CT, X-rays, and mammography, etc. After lunch, we got to go to the microbiology lab, where we learned how other techniques work, such as toxicology, and how drugs are being processed. And after that, we got to learn how DNA sequencing and PCR works at the core molecular lab.
So we spent our second day at the Neurology Institute, in which we were able to explore several specialties, including neuromuscular diseases, neuroimmunology, and the epilepsy monitoring unit. After that, we attended 3 seminars in which we learned about helping stroke patients, the brain's lymphatic system, and the biology behind the symptoms of dementia.
On our third day, we visited the Ophthalmology Institute, where in the morning we got to attend lectures from doctors who taught us about uveitis and glaucoma in the eye. For those of you who don't know, uveitis is inflammation of the eye. After that, we could see different methods that ophthalmologists have for eye testing and to revise that all parts of the eye are well and the vision as well. And after that, we got the amazing experience of attending cataract removal surgery and shadowing some surgeons, which all of us loved. And then after lunch, we got the chance to practice suturing on a model of the human eye.
On the fourth day, we were in the oncology and hematology department, where we had an immersive and incredible experience. So from the intricacies of radiotherapy to stem cell cryogenesis, we had an extremely fun time where we deepened our knowledge, especially on the apheresis unit and the blood bank, where we realized that although they're pretty silent units in the hospital, they play a very pivotal role in saving lives.
So getting to day 5, we went to nephrology and neurology. And this is like an overview of what we did. First, we did some surgical models practice, and we practiced suturing in a kidney. And we also got the opportunity to practice laparoscopic skills, which we later got to see at a bladder diverticulectomy surgery. So in this one, they practice and they use the laparoscopic techniques to actually perform the surgery, which were the ones that we were actually practicing before. And finally, in the afternoon, we went to a DNA extraction lab in which we could have the chance to extract DNA from a salmon sperm. And here you can see the DNA.
On day 6, we went to the cardiology department and we discovered the main branches of these fields, which are surgery, coronary care unit, heart failure, interventional cardiology, cardiac imaging, and electrophysiology and arrhythmias. Finally, we saw the diagnostic techniques, which are EKG, which stands for electrocardiogram, stress test, and echocardiogram.
Day 7 was our final day at the hospital. In fact, one of the most bittersweet moments for all of us. Our final day was at the respirology unit, where we got to make rounds at the respiratory ICU, the regular wards, and apart from it, even witness a catheterization take place. In the afternoon, we got to hear a talk from the Dr. Alvar Agusti, who offered us profound knowledge and deep sense of understanding of how research is really done. At the same time, also showing us how noble the profession of medicine really is, where we get the opportunity to serve medicine. But apart from the wards and wires, we lived a little. All of us were initially strangers, and eventually we became teammates, friends, and something more than family now. Some days we barely caught the bus, running to catch the last bus, and it turned to a low-key adrenaline rush. We explored Barcelona like locals, at times a bit lost, and on an average climbed 5 flights of stairs daily, which is something that was pretty dreadful, but at the same time one of the most memorable experiences ever.
I el punter deu estar al temps allà. El mouen ells, no? El punter? Sí. Ai, for me. Thank you.
Okay, hello everyone. It is my pleasure to introduce to you the Quantum Entanglement Group. So please give a warm welcome to Image, Sarah, Dan, Sofia, Emilia, Laura, Karma, Yuvan, Nicholas, Julian, Moose, and Rohan. It has been a pleasure, guys.
guys can pass the mic, please. Well, well, well. What do we have here? Ooh, a big box. Let me open this.
Freeze! This is Schrodinger's cat. Its life is determined by a quantum particle that decays, which causes a random collapse. However, the nature of quantum mechanics is that the cat is in a state of being dead and alive at the same time until we open the box. In other words, a superposition.
This is similar to how quantum particles behave in quantum mechanics. They exist in the same state of superposition until we measure it, so we can not know the state of the cat.
In addition, multiple particles can be entangled with another ones, meaning that the measurement made to one particle can directly affect partially or fully the state of another entangled particle. Entanglement can happen irrespectively of the space distance between particles.
Okay, anyways, why did I just freeze? Oh, I hope there's some money here. What the hell is this?
Breaking news! Last night, Schrodinger's cat was murdered. He was found dead. The cat was opened by Nick, and the cat's wave function collapsed to one state.
Listen, I didn't do anything. I didn't kill the cat. I just opened the box.
The courtroom erupted after Nick's lawyer, Niels Bohr, adamant to prove his innocence, and Albert Einstein collided, disagreeing on whether Nick should be tried innocent or guilty. Now, we will hear the declarations of both lawyers, Albert Einstein and Niels Bohr.
This man right here is innocent. He had no way of knowing whether the cat would be alive or dead before opening the box. There is mistake and injustice. Objection, Your Honor. There was a way for him to know whether the cat would have died before he opened the box. He had access to the local hidden variables, and therefore he knew the outcome of the box would lead to death. You're a liar! Shut up!
Order in the room. Bring the witness to the stage.
Heisenberg, I need you to tell me one thing. Do you know what happened on the night of the murder?
However, it's like some sort of Heisenberg uncertainty principle. But I found 2 sets of fingerprints on the box, which means someone else put the cat in there.
So, are you saying that before opening the box, there was no way to find out in which state cat would be in?
Of course, but what matters most is the true identity of the killer. If you'll allow me, I'll take the box into my lab for forensics.
This is absurd. The witness can barely hear us.
Enough. I have found a solution. I call it the CHSH inequality. Look. Oh, sorry, wrong slide. Anyways, if the variable is between positive and -2, then Einstein theory is right and Nick will be guilty. However, if it's between positive and -2 times the square root of 2, then Bohr's theory will be correct and Nick will be innocent.
We'll then perform measurements in different polarization configurations, and then we will compute the correlations between counts, photon counts in each of our detectors.
We can then calculate the value of S from the given formula using the correlation functions.
Then we went to the lab and we created a simulation with Python that matched the experimental results from the lab. And we found out we found out that the value of the inequality was 2.42.
I told you it's greater than 2. Shut up.
Breaking news! With the help of Bell's inequality, Bohr's quantum theory was finally proved.
Nick was found innocent because there was no way to find out if the cat was going to be dead or alive.
Thank you, lawyer.
Hello everyone, once again. I'm Laura Ruiz, the staff member of the Neurobiology group, and I'm so proud and excited to welcome this amazing group on the stage. Please, Sunida, Gabriela, Kira, Alice, Layan, Lindsay, Daniel, Isabella, Nuria, Lucas, Aina, and Tamara. The floor is yours.
So, as you know, for the past 2 weeks, every group has been working on their very own research projects, whether it was synthesizing paracetamol from scratch or building pocket cube satellites to send into outer space. But have you guys ever heard of mice getting PTSD or seen fluorescent scans of the brains revealing the most mysterious secrets of our most complicated organ?
In our Neurobiology of Trauma projects, we will explain our experience in the lab, using mice as models to understand how stress affects the human brain, looking at the effects of traumatic events and analysing behaviours in various types of experiments. Here we have our very own lab-bred experiment mice and their researchers. We hope you enjoy the video.
PTSD is diagnosed twice as often in women than in men. Why is that? Like human women, female mice also go through hormonal cycles that can directly alter their physiological, psychological, and emotional state. High estrogen and progesterone hormones enhance fear extinction, helping them recover from stress, while low levels are associated with increased anxiety-like behaviors. By straining tissue samples to track estrus stages, we were able to see which hormones were highest at that time. This is useful to know when to perform an experiment to show the most difference in behavior. By analyzing this, we can better understand why PTSD prevalence and severity differs so much between sexes and humans, and develop more targeted treatments that account for these variations.
Investigating the behavior of lab mice involves a range of tests which, in our project, were recorded and analyzed to highlight the effects of PTSD. We observed a strong correlation between the mice subjected to stressful experiences and reduced exploratory response, social interaction, as well as a large decrease in the motivation to seek pleasure, while anxious behaviors increased considerably. In response to severe stress, the brain reallocates resources to prioritize regions involved in survival, deprioritizing non-essential functions and causing the brain to react to normal situations in unusual ways. Transgenic mice are modified to have no genetic variation, so they are a stable framework for research. To observe the differences between them and non-modified mice, we can use some procedures, like doing a PCR. We extracted DNA, amplified specific regions, and used a thermal cycler so that the DNA could duplicate and become visible later. The last step was electrophoresis, which was used to separate molecules and proteins based on their size and electrical charge. Ultimately, this was scanned to create an image.
Immunofluorescence uses antibodies to label proteins like C-FOS, which gets expressed when the brain undergoes a stressful event. Antibodies are produced by the immune system and are designed to bind to specific regions of molecules. We first incubated the tissue with a primary antibody and washed it. Then we applied a secondary fluorescent antibody, as it allowed us to see the samples which determine which neurons are active or excited. We used epifluorescence and confocal microscopy to visualize the brain tissue. Both techniques used light to detect fluorescence. For instance, confocal microscopy adds lasers and pinholes for sharper, high-contrast images, allowing us to see the brain regions involved in PTSD, such as the amygdala, the hippocampus, and the prefrontal cortex.
We've been so privileged to study such an exciting subject using all manners of advanced equipment and facilities, even though we had to deal with the side effects of working with lab animals.
Hi everyone, I'm Andrea, and it's a pleasure to present the hardworking team of the Synthetic Biology Project. Please welcome to the stage Shaina, Cristina, Malika, Martina, Claudia, Isabella, Saud, and Gala.
Hi everyone. So today we're the Biosynthetic Club, and today we have decided to present our project as if it were a biological process. So, like in biology, our learning journey had some key parts. So the promoter to start, which is the introduction, the coding sequence, which is everything we've done, and the final product, which is our collective knowledge and skill.
Okay, so let's start with the promoter, which is where we consider our expression to begin. So to start, we would like to introduce you a bit to the world of synthetic biology. So basically, if you can look like at the white circle, synthetic biology takes its inspiration from life's natural blueprints, such as DNA, and it uses it to modify, design, and build new biological functions with purpose and precision. So, despite all of this, nothing would be possible without the presence of a very, very small, yet crucial element called a promoter, which, well, if we don't have a promoter, expression would never be possible. And with it, well, everything begins.
Okay, so next we have the coding sequence. So the coding sequence contains all of the instructions that is needed in biology, right? So those instructions are written in the codons, and these codons determine the shape of the nucleotides and the proteins and the function that serves. And so, like the coding sequence, we will go step by step.
On the first day, we covered the basics of synthetic biology, genetic circuits, modeling, and genetic engineering.
On the second day, we had a wonderful experience of coding our own plasmid using Benchling, which is a coding website. And then we performed the structure in the lab when we created our own plasmid using Gibson Golden assembly, and then inserted our plasmid in E. coli and get a bacteria.
Okay, so the next day our bacteria was ready. So we picked 7 colonies at random and to insert into a PCR mixture, right? So we conducted a PCR polymerase chain reaction in order to amplify the plasmids that we wanted. Afterwards, we loaded the PCR mixture into an agarose mold gel and then performed electrophoresis. This determined which colonies had the inserts that we wanted and which colonies didn't.
On the 4th day, we extracted our plasmid, did ONT sequencing, and did genetic circuits induction.
On day 5, we had a lecture on arts and synthetic biology, drew with fluorescent bacteria, and learned about CRISPR/Cas9 gene editing technology.
On day 6, we learned about fluorescent bacteria drawing results, and then we had another CRISPR/Cas9 lecture, and then we also had time to work on our projects for a poster session, which I'll be explaining on the next slide, and then we had an ethical debate based on synthetic biology practices. For day 7, we had our poster session. This is where we presented unique ideas on ways to utilize synthetic biology to solve problems of our own choice, such as ways to facilitate type 1 diabetes and solving gray hair and many other problems.
Okay, and finally, for the last part of the presentation, the final output, which is the expression of our protein. So basically, expression is when the invisible becomes visible. So basically, when DNA becomes RNA, RNA becomes proteins, proteins become function, and function becomes life. And instead of looking like this, our protein ended up looking a bit more like this. So we have prepared a musical parody called Genes That Make You Beautiful, so we hope you enjoy it.
You're a codon, don't know what's on. You're turning heads when you create a mutation. Don't need Cas9 to be alive. Being the way that you are is divine. Everyone else in the lab can see it. Everyone else but you. Baby, you light up my world like fluorescence. The way you dance and let me overwhelmed. But when you promote that sequence, it ain't hard to tell. You don't know. Uh-oh. Genes that make you beautiful. And when you saw what this can do, you'll understand why I want to learn about you. And now I'm looking at your criteria. If you don't know, uh-oh. Genes that make you beautiful. Uh-oh. That's why genes are beautiful.
Hello, everyone. Thank you. Well, I wait. I'm Gaia, the staff member of the crack group, or also known as the Melon team. I'm very happy for these 2 weeks. Thank you guys for the great team we've built, and I'm very proud of what you achieved and what you learned. So please welcome to the stage Andrea, Leonor, Eamon, Clara, Jodalis, Lisa, Isabel, and Ruben. The floor is yours. Juan could not be here today, but he was also part of the group and left his contribution via a video.
Or not to be orange. Hey Biscuits, have you ever thought what changes Melon's flesh color? Here at Craft, we've been studying this question for the last 2 weeks. Let us tell you about it.
But why orange? Most recent vegetables with orange flesh have beta-carotene, a carotenoid that the human body converts into vitamin A. This nutrient has a lot of benefits, such as improving your eyesight, hence why your parents might have told you when you were a kid to eat more carrots to help you see better, because mine did. The goal of this project was to find ways to modify the genetics of the most popular variety of melon, Piel de Sapo, and turn its flesh orange to give it a more nutritional value and more vitamin A, like this one.
So, to crossbreed plants, you need the parental lines. In our case, was Vedrante and Piel de Sapo. So, to cross, you need to put pollen from the male flower into the female flower, like I said, pollinating it. With the offsprings, we first need to check their visual traits, and this process is called phenotyping. We phenotype a large list of traits, such as size, width, sugars, and so on, but the most important one for us was the flesh color.
Genotyping means analyzing DNA to check difference between two individuals. We use it to find plants look like Piel de Sapo, while with the oral flesh color like Vedrantes. In DNA extraction, we start extracting DNA from young leaves. This gives us the genetic material we need to study. A molecular marker is a marker as specific DNA sequences. This uses to find traits inside the genome.
Genetic mapping and QTL analysis are used to identify regions of DNA linked with certain traits, which in our case is the flesh color in melon. We collected genotype and phenotype data, and by comparing them, we pinpointed a location, as you can see on the graph, where the DNA is strongly associated with the orange color in the flesh of the melon. The peak of the graph shows the QTL, which contains hundreds of different genes, and that's where the gene that we're looking for would be found.
We made a table with the 9 melons we work with and the parentals. We have genotype chromosome 9 with 7 markers called SNPs. In the table, we can see the alleles of each melon in each marker to check if they have the piel de sapo allele or the verberante allele. This way, we can know which one is associated with the orange color gene. If we take a look to the SNP 6, we will notice that every time it has the piel de sapo allele, the melon ends up being white.
So now that we more or less know the specific area of where this gene could be, it's time to narrow it down using 2 positions, as you can see on the screen. When we look at the first graph, we can see that the small region in chromosome 9 contains only 11 genes. So in order to identify which gene is responsible for giving the melon its orange flesh color, we researched in the melonomics database, and we were able to locate one orange gene that perfectly matched the orange flesh phenotype.
In order to validate the gene that gives the orange color, we have used the technique of CRISPR-Cas9 in orange fresh melons that consists of cutting the gene that we have identified. If the gene that we discovered really controls the fresh color, then the edited verberante melon will have white flesh. If the melon turns orange, it means that we have cut the wrong gene. Through the CRISPR-Cas9 technique, we obtain mutated plants that we put grow in vitro.
Through our extensive research on molecular breeding and gene editing, we have successfully identified and specified chromosome and genome regions responsible for flesh melon color. This breakthrough will not only advance our understanding of melon genomics and genetics, but also pave the way for more targeted breeding strategies. We would also like to express our sincere gratitude to CRAG in Barcelona for their invaluable support and collaboration in making this project a success. Thank you. Sorry, sadly Juan couldn't be here, so we made this for him. He's in Amsterdam.
As we arrive to the end, we are going to finish very high. We are going to finish at the space. In an industry that is growing on the world, in Europe, and here in Barcelona, thanks to Nanosat Lab. So, for explaining this 7 days project, please give a warm welcome to the stage to Arnau, Cyrus, Arman, Rafael, Daniel, Soyun, Sophie, Mikel, Paul, Gonzalo, and
hello. Well, hello everyone. We're the BIYSC satellite group, and in this presentation, we'll talk about how we made an actual satellite that can take pictures of Barcelona from the sky. So first of all, what are pocket cube satellites? Well, they're a type of
picosatellites, meaning that they weigh no more than 1 kilograms, and they size only 5 times 5 centimeter cube. And they're normally made for educational purposes, as they're, again, really small and cost-effective comparison with other type of satellite like CubeSats.
Our mission is to create a pocket cube satellite that is capable of taking photos of Barcelona from the space and sending them to the ground station.
Yeah, but like before building the satellite, we have to know what orbit we're choosing, and we have chosen the sun-synchronous orbit. So basically it flies along with the sun. So the benefit is it can charge with the solar panels. And then this is not enough because we need more data, and we use the MATLAB to calculate and simulate the orbit of the satellite. And this gave us the eclipse time. So we can use this to calculate how many power we need, the size of the battery, and this allows us to know the power budget. And we also know the access time. So basically the time where the satellite goes across the ground station for data transmission and receiving. So we can therefore know the link budget and the data budget.
So to be able to construct this satellite, we need to follow a step of procedures and things we need to do. And here you can see all the list of things we had to do to assemble the hardware.
As the second part of the work on a satellite, you can see the software, and there is functions of our satellite now.
Now here is an actual 3D model of what our satellite should look like, and it has the payload and also all these other PCBs, which are essentially printed circuit boards, the electrical power system to provide proper power to every single part of the satellite, the ADCS to determine orientation and position, and the OBC you can think of like the motherboard of the entire satellite. Now the payloads are essentially what we talked about, the stuff to actually get the purpose of the satellite done, such as the camera, the GPS, and the EMU. We also have the solar panel on the side, which helps with electrical power.
So regarding the software part, we programmed our satellite in the C programming language under a free RTOS operating system, and we basically programmed the telecommands as well as the communication between the inner devices of the satellite. And the procedure we followed was the following. We first designed how we wanted our satellite to work, and then we implemented the code. And we compile it and run it in a qualification model. We debug it with special STM32 tools to find that other bugs have appeared. So then we went back to the first step, and we repeated the process until the satellite worked, behaved as we wanted.
Okay. As for the actual manufacturing process of our satellite, we 3D printed some of the outer structure of the satellite, as you can see in the picture. And then to make our payload PCB, we used an ECAD software called KiCad to design the schematic and decide our component placement for our payload PCB.
Once we had the PCBs ready, we had to solder all the electronic components onto them. This was a pretty complicated process because it involved putting together very small electronics, some of them like average of the size of a grain of rice, on a tiny little plate using a tool that is on average the size of a pen. So this was very complicated. And once these complex electronics were assembled, we put together all the parts of the satellite inside of the clean room, which is a special sterile environment that is free of any kind of dust that could ruin the electronics and contaminate the satellite.
Okay, so what we used these software engines, for example, SolidWorks, to design and modify our pocket cube. The picture of it you see previously. KiCad to design our PCBs, and at least not the final but not least, Radian to simulate space environment and see our satellite's thermal condition. They didn't pay us for advertisements, so we're still waiting for it.
Finally, after having all the parts ready, we did the final assembly inside of the clean room, and we had a functional pocket cube. We did the full functional test and verified that every function, every part of software and hardware worked, and therefore verified that it is a functional pocket cube. We would like to thank the staff, the researchers, and all of you. Thank you very much.
Well, and that was the last presentation, actually. So congratulations on what you just did. We think that all the projects you just presented are super relevant. You worked very hard on them. All the insights, the results you showed are really amazing. And you communicated all of this in a very rigorous, but at the same time, funny way. So I think you should be very proud of yourselves. We are very proud of each of you, seriously. And now, before we wrap up this 2025 edition of BIYSC, let us express our deepest thanks. So first of all, to the researchers, the research centers, institutions, hospitals, the teams of scientists and professionals who have worked hand in hand with the participants during these days and whose contributions and dedications are just crucial to make BIYSC possible. So once again, thank you very much for all your collaboration. It's an honor to do BIYSC with you. What else, Lucas?
Yeah, and BIYSC would definitely not be possible without the staff team. You have been truly amazing. You have taught us that science is not only research, it's also about empathy, generosity, teamwork, and community. Thank you. It would not be possible without you.
Also, a very special thanks to the co-coordinator of BIYSC, Monica Chauen, and to everyone in the team of Science, Education and Research at Fundació Catalunya La Pedrera for the invaluable support here today and always.
And let's also thank all the families that are here today and those that could not come today because they're rooting for you every day and they trusted you to come to the program and they trusted us. So thank you very much as well. And then, last but not least, thank you all participants because you have created the best environment we could have ever hoped for. You have been truly amazing. You learned a lot. You helped at all times. And I think we all enjoyed being with you. So thank you very much. Now your journey continues, but remember that you have your peers, you have the BIYSC staff team with you, and you have the Science Fellows community to support you throughout your career.
And we hope to see you again in some time. And now, before we finish, we have a final special surprise for you. So let's look back to these amazing last 2 weeks. Here at BIYSC, participants get to work in real research settings. They are experiencing how daily life in science is really like. I mean, they're building nanosatellites, they are researching how trauma affects the brain or how to genetically edit a crop for it to be more resistant. So they get to work hands-on in very cool projects that at the same time they address real and very relevant challenges.
It's really cool, it's really exciting.
I never experienced really that in school because we have all different interests and here we're all here for sciences.
We're connecting a device to identify the light.
So it's kind of interesting to see how their education path was different, but it still led them here to BIYSC.
I really want to do something more sciencey, like in my future, because like I really want to become like a physics person myself. And I want to try to understand that I really like it, and I want to like confirm this information by BIYSC.
The atmosphere is actually amazing. We're very lively, everyone's getting along with each other. They're very friendly, the researchers as well. They make sure that we understand what we're working on. They always encourage us to ask questions.
I've never seen an application of such high-level maths until I came to this course. It's really given me an insight into what I want to do in the future, and I think it's honestly a great opportunity.
We were basically having a deep dive into all the various departments of the hospital clinic. So over a span of 2 weeks, we explored various different wards and departments.
I've been able to meet different people from these different ethnicities and different countries, and it has been a pleasure to meet everyone.
This is like the first time I've managed to be in a lab and actually participate in research, so it's made me want to go more into what I want to go into, so yeah.
I'm participating in is making a pocket cube satellite, which is a small satellite that's made by a standard convention called pocket cube. We're learning about the different components in a satellite, how to determine the orbit of the satellite, and how to manufacture it, including how to solder, run structural and thermal analyses.
I think this experience gave me a chance to see how people work in the aerospace field.
And what makes all of this even more exciting is the BIYSC community that is created here. I mean, look at this, over 30 nationalities coming together, learning from each other, and building skills that go way beyond science.
It's my first time in a professional lab, and I love the experience.
And I think the most important thing is that they know we're not their level, so they know how to maybe simplify a bit the topics so that we understand, or else it would be very complex.
I really loved the walking tour and just getting to really immerse myself in the city and to look around at the beautiful architecture.
I feel really privileged to be in this program, as it's really fun and challenging, and it's a good balance between theory and practice.
I think after learning everything, I became much more confident. But with quantum mechanics, you can never know everything.
The participants are being accompanied by brilliant staff members that accompany them through this amazing journey, through going with them to the projects, helping them.
BIYSC staff and coordinators were so kind and so generous.
I have some of them, they are my friends, and I love it.
I think this is a pretty good opportunity, because I do know a lot of people that might go oh, staying in the lab all day might not actually be what you want.
The people here are incredible. Everyone here is so nice, and I've made a great group of friends from every country on earth.
I think this project is really a mix between architecture and engineering, two fields that really fascinate me. The involvement of actually engineering and mechanical constraint, line constraint, in the design of the La Pedrera by Gaudí, and how he really thought of all of those elements in his conception of this beautiful piece of work.
We're a really small group, so I feel our connection is very strong. It's so much easier to communicate with each other and have more attention to us as a smaller group.
I think that I enjoy the practical part the most, because it's the kind of skills that you need to apply for a job.
For me, the thing that I enjoyed the most was when we did clinical cases, but they were simulations. So we got a patient, or a name and some data, and then we had to try and figure out what the diagnosis was. And we had some really interesting discussions.
Today we are painting with bacteria, and you might be oh, you can't see anything. Well, that's because it's fluorescent. So you need a special light, like a black light, and also to wait for the bacteria to grow for 24 hours to actually see my design. We've been learning about CRISPR-Cas9 technology and the basics and the complexities and anything to do with gene editing.
I think it's a really great experience. Everyone is very welcoming.
I'm growing both scientifically and socially.
I would absolutely recommend BIYSC to anyone with a passion in science.
If anyone is thinking about applying or not, do it. Just do it because it's an amazing experience and you will have a great time.
Thank you everyone for attending. Please, the staff members and participants can go back to the residence and we'll do the checkout from there. And the rest, thank you and have a good summer.